It's an organized cargo that can be transported by a single genus particle motor. So we have all this capability, and now we'll see application from environmental, defense, biomedical, energy generation using this capability. But the initial focus was biomedical, and here we have several challenges on drug delivery, biopsy, surgery. One challenge is we have to address the toxicity. We cannot use our peroxide fuel. We need to look for alternative fuel. And also this nanowire cannot operate in high ionic strengths of the blood. We need to look for new motor, new propulsion mechanism, not self-electrophoretic. So we are moving to a new one, which is our micro rocket, which are bubble propelled. This can operate efficiently in biological media and high ionic strengths. Also have a lot of power by the bubble truss and can be functionalized. So this, as you'll see, these are open tube, conical microtube bilayer with the inner layer of platinum, where the peroxide fuel is decomposed, generate this oxygen bubble that propel it as a thrust like any other rocket. So we prepare them by, again, template electrode deposition. The membrane is a conical micropore, so we have this conical shape essential to the evolution of the bubble from one side. You see the outer layer is a polyaniline, the inner layer is a platinum, and again the peroxide will decompose internally, generating the oxygen bubble on one side. So these are very powerful, very efficient. Again, this example of the preparation, again 5 micrometer long, 1 micrometer diameter. And this, after you dissolve the membrane, you get many of these. And again, you see the movement. These are 200 millisecond interval. You see the bubble truss in the next video. You'll see a remarkable speed of nearly 400 body lengths per second. So if this is 5 micrometer, it will go 2 millimeter per second. And 400 body lengths. So 5 micrometer go to 2 millimeter, like you can see here. This is a slower motion. And these are guided magnetically. We also have a magnetic layer. So this is the oxygen bubble truss with high speed and power. I mean, you have remarkable flower type. You can do all type of, depend on the way the bubble are evolved. But if you look at this speed, we actually now have over a motor with over 1,000 body lengths, which is 10 millimeter per second. This is the if you compare it to other fast objects, like the cheetah, the faster animal, the cheetah go only 20 body lengths per second. It's a lot of kilometers per hour, but only in body lengths is much slower compared to my rocket. Or your Ferrari is only go 20 body lengths, or the bullet train will go even slower in time. So this is really the faster object. Actually, there is a journal of Wiley chemical record that shows this capability. It's not only fast, it also operates in high ionic strength media. This bubble propulsion is independent of the ionic strength. You see a comparison with no salt and with lot of salt. This is again slow motion. You can see that the speed is about the same in the high ionic strengths. We can operate in biological media. It's a little slower because of the viscosity. You can see in a 1-to-1 diluted serum, they swim nicely. Also, when you functionalize them, you can do this collective behavior of multiple rockets, coordinated, magnetically guided in a collective fashion. So we have all this capability. Now we can go to the microchip. We can try to isolate biological target like anthrax or cancer cell, basically putting a receptor on the outer layer to capture, transport, and isolate it from untreated samples. So over the year, we introduced motor with different receptors like single-stranded DNA or antibody or aptamer. We control the surface chemistry on the outer gold layer. We have self-assembly monolayer. This mixed monolayer minimize non-specific adsorption. And then we can do on-the-fly hybridization or protein immunoassay directly from untreated raw complex samples. So this example, 2011, we show we can capture, transport, and isolate cancer cell, pancreatic cancer cell, by putting an antibody selective to the antigen expressed on the cell wall of this pancreatic cancer cell. This is very selective, and you can see, first you can see that this moving with the antibody is efficient in the human serum. So the modification is not influencing much the propulsion, and then we can use it to capture and isolate the cancer cell. This is in a buffer, this is in serum. You'll see in the video it approaching the cancer cell, contacting and transporting it, as you can see. Don't know if you can see the cancer cell here, and the motor is approaching. Now it's picking it, transporting it, and release. This is now in the next one is in serum. Do the same and isolate the cancer cell, which is important for cancer diagnosis. For DNA hybridization, again, single-stranded DNA, we have sandwiches with a fluorescent probe. You can visualize it, go to a very complex samples, and again, the mixed monomer, and you'll see the fluorescence of the probe. Only when you have the target, this is excess of a mismatch or excess of non-complementary, no fluorescence. Only when you have the target, you have sandwich with capturing the fluorescent probe, like in this image. Only with the target, you see the complex sample. Again, you see the control. There is no fluorescence in this excess of control, mismatch, and non-complementary, but only in the presence of the target. And we took it for RNA detection from bacteria. Again, this is the target bacteria, low level. This is a non-control bacteria in excess. Only where we have the target, we can see the fluorescence of the RNA hybridization. We recently applied even for intracellular mRNA detection, where we have the motor, which is based on ultrasound. Ultrasound, they can pierce a cell and penetrate the cell, release a DNA probe to hybridize with the mRNA in the cell. And this release because here we have initially the probe is with a dye. The dye fluorescence is quenched when it attached to the gold and the graphene layer. Once it released by the hybridization, you have a fluorescent on. So you can detect the mRNA from the fluorescent signal. This is all intracellular detection that we just published, ACS Nano. So again, penetration into the cell, moving into the cell, and they accelerate hybridization due to the movement, lead to release of the probe, and the quench fluorescence is disappear, and now you see the fluorescence on. We can also do aptamer to transport, detect protein like thrombin, also to release the drug, thrombin drug on demand. So this is a mixed aptamer attached to the gold coated layer. It can capture the thrombin, and then you add ATP to release the aptamer, and you can release the thrombin from the surface. Bacteria isolation, this is E. coli, where we have lectin on the surface that's moving around. This is a big DARPA project. We aim to capture bacteria, anthrax, other biothreats. So in this case, you see the bacteria in drinking water, and you see the motor with the lectin approaching the bacteria. It's 2 micrometers. You can visualize it. This is an apple juice, so different swimming, but it's still all selective. In seawater, I think next. And you can visualize it because the motor is 5 micrometers, the bacteria is 2 micrometers. So all this capability allow us now to go to lab on a chip, and you have a complex sample where you can capture the bacteria or a cancer cell. So have the motor with the probe enter the sample compartment, reservoir, capture, transport, isolate, a different reservoir to clean up for enrichment, detection, and then regeneration. So all this lab on the chip allow to send different motor with different receptor to pick up different target. Then you can transport them, isolate, enrich them, detect and regenerate the motor for new application. So we demonstrated this initially with the antibody immunoassay, where we have anti-IgG to detect IgG protein. So we have a polymer outer is a carboxy PDAT. You can functionalize the antibody by EDC coupling. You have the inner layer of nickel for magnetic guidance, and then the catalytic layer. So the motor will go to the sample reservoir, selectively pick the IgG target, transport it, and you'll visualize it with a microsphere you'll see in a moment. So the question, can you move this antibody in complex channel? Can they go to the sample reservoir and carry the antigen? So you can see here, it's swimming in the channel, they go back and go to this side or this, will go in the reservoir, if you see again, all in 2 micrometer channel, again 5 micrometer motor, 100 nanometer diameter. Here it go to the sample and pick the target and leave the sample reservoir, and swimming, it's more viscous in the blood, so it goes to follow up application. And you can go to the second reservoir where the secondary antibody tagged with a sphere, and you can visualize this sandwich assay under the microscope directly, just because the sphere is 1 micrometer. So only when you have the target you can see this sandwich assay formation. Again, 5 micrometer motor, 1 micrometer sphere for direct visualization. We can also have motor with built-in recognition without a receptor, the motor itself acting as a receptor. If you use motor which are built on boronic